Hello, welcome everyone to this special edition of UMDF's Bench to Bedside webinar series. Today's topic, mitochondrial replacement therapy, shared experiences from the UK and Australia, and implications in the United States. I'm Philip Yeski, Science and Alliance Officer at the United Mitochondrial Disease Foundation. I'll be your host for today's meeting. Before we begin, though, just a couple of background slides. One, we always like to call out MitoShare, a patient-populated global registry for mitochondrial disease patients and caregivers. If you're a clinician who is taking care of mitochondrial disease patients or a member of the community, we encourage you to join. It's only through having you engaged in clinical research that we can advance the therapies that we know are necessary for mitochondrial disease. Also like to mention that Combined Brain is our partner for a biorepository. So please contact us if you're interested in providing a biosample to the repository. Also, just a reminder that UMDF hosts an annual scientific conference, and next year we will be in Orlando, Florida. Consider this a save the date, June 17th to 20th, 2026 in Orlando, Florida. UMDFconference.org is the place to find out all the details. We're putting a great scientific and medical program together. But as most of you probably know, we also run a family program. Please consider joining us. It's a great way to network with all the stakeholders in our community. All right, with that behind us, thank you again for joining for this really critical discussion. We're here today to address a profound unmet clinical need, the prevention of mitochondrial DNA diseases. As all of you who are on this call are aware, these are devastating disorders for which there is no cure. There are countless stories in our community of mothers enduring pregnancy losses, families losing children to Lee syndrome, my own family included. All of this just underscores the deep urgency of our work. Against this backdrop, mitochondrial replacement therapy has emerged as a beacon of hope. This innovative expansion of in vitro fertilization offers women the opportunity, women who carry pathogenic mitochondrial DNA, to have a healthy child that is 99.9% genetically related to them and their partner and effectively stops the transmission of the disease from mother to child. However, the path to clinical use has varied dramatically across the globe. In the United Kingdom, which stands as a model of progress, through a deliberate decade-long process, scientific review, extensive public debate, the UK has successfully built a consensus and trust needed for parliamentary approval, and that arrived in 2016. In Australia, at the time of approval, it was not legal. Approval in the UK was not legal, but it was recognized that mitochondrial donation does afford this opportunity to allow impacted Australians to have genetically related children without the risk of inheriting mitochondrial DNA defects. The eventual passing of Mave's Law in 2022 set the course for clinical research and will allow MRT to proceed in Australia. We'll hear a lot more about that today as well. However, in the United States, the situation is starkly different. And while the National Academies of Sciences, Engineering, and Medicine concluded in 2014 that clinical investigation of MRT are ethically permissible under specific conditions, progress is at a standstill. A congressional rider currently prohibits the FDA from even evaluating clinical trials using this technology. This legislative roadblock exists despite the clear scientific and ethical support from the nation's scientific body, leading scientific body, and the desperation of families. This has led some to seek unregulated treatment abroad, a situation we do not find acceptable. That's why we gathered today. Our goals for this meeting are threefold. First, to hear from our expert speakers in the UK, Australia, and the United States on the current scientific and clinical status of MRT. Secondly, to learn directly from our patient advocate colleagues in the United Kingdom and Australia about the pivotal role they played in achieving government and public support in advancing MRT to the clinic. And lastly, perhaps most importantly, really set the stage to use these collective experiences and lessons to inform our path forward in the United States. This meeting is the first step towards convening US-based patient advocacy groups for a follow-up strategic discussion on what can and must be done to advance this field in the United States. We have a unique opportunity to learn from those with experience in this field today, how they've navigated this complex terrain. I want to thank in advance our speakers and panelists for their contributions to today's meeting. And with that, I'm going to turn it over to Dr. Michio Hirano, who will serve as the moderator for the first portion of our meeting today, the clinical experience with MRT. Dr. Hirano.
Okay, thank you, Dr. Yeski. So it's my pleasure actually to participate here today, and we're very fortunate to have two of the major authors on the two reports in the New England Journal 2 months ago talk about their experiences in Newcastle, and I will introduce our speakers. First speaker will be Professor Bobby McFarland, who's based in Newcastle in northeast of England, where he's the director of the NHS Highly Specialized Service for Rare Mitochondrial Disorders. Bobby is a pediatric neurologist and well recognized internationally. He provides a clinical service for children with mitochondrial disorders. He also leads a mitochondrial reproductive options clinic and is research lead on the follow-up of babies following mitochondrial donation. Maybe I'll go ahead and introduce our second speaker because they'll speak sequentially. And actually, Bobby will speak first, and then Professor Herbert will speak, and then Bobby again. So the second speaker will be Professor Mary Herbert, who is a reproductive biologist in her role as a professor of reproductive biology at Newcastle University and scientific director at the Newcastle Fertility Center, where she established a closely integrated team of research and clinical scientists, including Dr. McFarland. In this capacity, she led the team that developed pro-nuclear transfer, PNT, to reduce transmission of mitochondrial DNA disease. She's the lead author on one of the two New England Journal articles reporting reproductive outcomes of a clinical pro-nuclear transfer and pre-implantation genetic testing program for mitochondrial DNA diseases at Newcastle. Recently, Mary joined the Monash University in Melbourne, actually in July of 2023, where she leads an MRT research program as part of an Australian government-funded $15 million Mito Hope initiative to test the feasibility of implementing a clinical MRT program in Australia. So these are two esteemed speakers, and I'll turn it over now to Professor McFarland.
Good. Okay. Well, first of all, thank you for the invitation to speak about our work recently published in New England Journal of Medicine. As Michio has said, Professor Mary Herbert is going to talk about some of the embryological aspects of that work, and I'm going to speak about the reproductive care pathway and then subsequently on the follow-up of children born after Mary has spoken. So I understand that this is a fairly broad audience, and so therefore I think it's sensible to begin with a brief overview of mitochondrial DNA-related disease. Hopefully this will be a refresher for most people. So mitochondrial diseases can be devastating multi-system disorders that occur anywhere on the age spectrum, famously described as occurring in any organ and at any age. Mitochondria are the powerhouses of the cell and are under the direct control of two different genomes, the mitochondrial genome and the nuclear genome, with a total proteome of around 1,300 proteins involved. Today, though, we're going to focus particularly on the mitochondrial DNA in relation to disease prevention. So mitochondrial DNA is maternally inherited, but a genetic bottleneck means that the inherited heteroplasmy load may be quite different between siblings and different in those siblings from their mother. Mitochondrial DNA is multi-copy. Disease is typically associated with heteroplasmy, where a proportion of mitochondrial DNA molecules are mutated. When the proportion is above a specific cell threshold, then dysfunction and disease occur in that cell and subsequently in the tissue. In some circumstances, this threshold is extremely high, and disease only occurs at homoplasmic levels of the mutant, shown in red here. For heteroplasmic mitochondrial DNA disease, it is generally possible to offer pre-implantation genetic testing or indeed prenatal diagnosis through something like a chorionic villus sample or amniocentesis. But what about reproductive options for homoplasmic mutations? One such case is that of Sharon, whose family formed the basis of my PhD some 25 years ago. Sharon was relatively unaffected, but as you can see from this family tree, 6 of her children died actually in the neonatal period, and a 7th child died in early adult life from a condition known as Leigh syndrome. So it was in relation to Sharon and families like her that Prof. Turnbull and Herbert took on the challenge of providing an option for women to have children that were genetically their own and not pass on these dreadful mitochondrial DNA-related diseases. I don't think either of them thought at that time, though, that there would be a more than 20-year odyssey and involve 4 separate scientific and bioethical reviews, as well as an act of parliament to get to the stage where we could actually offer this in clinic. That was the pathway that we had. I'm sorry, have you not heard that?
No, we hear you fine, Bobby. Thank you.
Oh, sorry, it flashed up a message on my screen. Sorry. This is Jane Ellison, who's signing mitochondrial donation into law following an act of parliament. She did that in March 2015. But it was a further 2 years before a UK license for mitochondrial donation was issued to Newcastle, allowing us to actually begin the process of performing MD. So we had to design a reproductive care pathway that allowed us to implement this technique safely and cautiously. That involved assessing the fitness of women coming forward to carry a pregnancy, and we devised a mitochondrial reproductive advice clinic specifically to do that. Since 2017, this clinic has received referrals for 196 women, and 169 have been assessed. 131 of those have subsequently been referred on to the Mitochondrial Assisted Reproductive Technologies Clinic, otherwise known as MART. The MART clinic involves a much more detailed discussion of PGT and mitochondrial donation, as well as an assessment of the male partner's health and sperm analysis. At this clinic, we obtain consent for a research program and establishment of threshold heteroplasmy in the cases of PGT. So we have to establish a level that everyone is happy. It goes back in the cases of pre-implantation genetic testing. So of the 131 women referred, 70 women were offered either pre-implantation genetic testing or mitochondrial donation, with 2 couples having both techniques performed. 32 HFEA approvals have been obtained. Each couple have to apply independently for HFEA approval. That's irrespective of whether the mutation for which they are asking for mitochondrial donation has been approved previously. So we have to do an individual application for each couple. We've performed PGT on 28 women and had 13 live births, and Mary will talk a bit more about this. We've had 22 mitochondrial donations performed and 8 live births as a result of that. Okay, what I'm going to do is I'm going to pause at that point and come back after Mary has spoken about the mitochondrial donation embryological aspects.
I'm going to talk about the reproductive outcomes paper from our ART treatments to reduce transmission of mitochondrial DNA disease, published recently in New England Journal, and Louise, who's on the call, was first author on this paper. Okay, so for you, those of you who are not familiar with this, I'm going to first introduce you to the key players. At the center of the screen here, we have a human fertilized egg. And in that egg, we'll see 2 large structures called pronuclei. And the pronuclei contain a single copy genome, one inherited from the egg and the other inherited from the sperm. Sorry, I'm having problems here with this. But the egg also contains about between 200,000 and 500,000 copies of mitochondrial DNA. And this is inherited exclusively from the egg. So as Bobby explained, mitochondrial genome is maternally inherited. The nuclear genome is inherited from both parents. Then over the course of 5 to 6 days, this fertilized egg undergoes a series of divisions, eventually forming a blastocyst. And the blastocyst is an important developmental milestone because it is the stage at which an embryo can implant in the uterus. And then over the next 9 months, it will go on to form a baby, all going well. So what I'm showing you here is a fluorescence image of a human egg. And the red marks mitochondria and the green marks the DNA. And you see the nuclear DNA here. This egg is poised for fertilization. But throughout the egg, there is the mitochondrial DNA as well. So what you can see from this is this egg is just jam-packed full of mitochondria. And for women who carry pathogenic variants, they can be present in some copies, and that's called heteroplasmy, as Bobby explained. Or if the mutation is present in all copies of mitochondrial DNA, it's called homoplasmy. So the assisted reproductive technology treatments to reduce the risk of mitochondrial DNA, the first one is pre-implantation genetic testing. So this is a treatment that has been used for decades in ART labs to reduce or to prevent transmission of nuclear encoded genetic mutations, but it can also be used to detect mitochondrial mutations, and that involves culturing the egg until the embryo divides, and then removing a cell. Either we do it at the 3-cell stage, could also be done at the blastocyst stage. And these cells can be then sent to the diagnostic lab to measure the heteroplasmy level. And then that enables us to identify embryos with the lowest mutation loads for replacement in treatment. And so this is our paper reported our work on PGT and PNT, which I'm going to talk about now. And an interesting thing is that, as far as I understand, PGT is not available in the US either. So for those women who are not suitable for PGT, so the success of PGT depends on the availability of embryos with low mutation loads, and that's not always the case, especially, of course, if their mutation is homoplasmic, we can't use PGT. So for this, the idea of mitochondrial donation or replacement was developed. And it involves transplanting the nuclear genome to an enucleated egg from an affected donor, and it's suitable for women who are likely to benefit from PGT. And it can be done before fertilization, and that's called maternal spindle transfer. Here in this drawing, you'll see the spindle. This egg is ready to be fertilized, but we can remove that spindle using special optics. but it can also be done after fertilization by transferring the pronuclei. And the procedure we used in Newcastle was pronuclear transfer, simply because most of the animal studies had been done on this at the pronuclear stage at the time we started out. So we have optimized the pronuclear transfer procedure over a number of years, first publishing proof of concept in 2010 and later publishing preclinical research. And it involves collecting eggs from the donor and from the patient. These are fertilized with patient partner sperm normally. Then the pronuclei appear and they are removed. The donor pronuclei are discarded. The patient pronuclei are fused with the enucleated donor egg to give us this reconstructed zygote containing the nuclear DNA from the affected woman and her partner, and predominantly the mitochondrial DNA from the donor. And I say predominantly because when I showed you that egg earlier, you understand what a challenge it is to extract this nuclear DNA without any mitochondria. So when we fuse this the nuclear DNA with the enucleated egg, it will be surrounded by some mitochondria from the patient egg. And that is a limitation of this procedure that we'll come back to later. So this is Louise performing pronuclear transfer. So removing one pronucleus at a time. And then they're taken very briefly and exposed to fusogen, which is an activated Sendai virus. And then they are fused very quickly with this enucleated donor egg. So as I said, that procedure was optimized over quite a period of time. So just to talk you through the clinical interventions that patients want to go, either PGT or PNT. So we have, for standard IVF treatment, we get ovarian stimulation that involves hormone injections to stimulate growth of multiple ovarian follicles. And then once we have sufficient follicles, after about 11 to 14 days of injections, the eggs are retrieved by transvaginal ultrasound guided aspiration. And then these eggs are fertilized, usually by ICSI, that involves injection of the sperm directly into the egg. And I say usually by ICSI, it's usually by ICSI for the purpose of PGT or PNT. And then these embryos are cultured, and for 5 to 6 days, they become blastocysts. The best quality blastocysts are transferred to the uterus, embryo transfer, or if there are additional blastocysts, these are frozen by vitrification and transferred at a later date. So the additional steps for PGT treatment is to biopsy the embryos to remove a cell. We do it on day 3, and that cell is sent for analysis. So that we then select blastocysts, the best quality blastocysts with the lowest mutation loads. And the additional steps for PNT is that we do the egg retrieval and we freeze all of the patient eggs. And we stockpile them so that we can, until a suitable donor becomes available. And then once the donor is available, we thaw these eggs and we fertilize them using the patient partner sperm. So these are the additional steps for PNT. And so far, in our program, or at the time of publication of the paper, we had 39 women had undergone ovarian stimulation for PGT, and 39 had had an egg retrieval, 39 had ICSI, 37 had embryos suitable for biopsy, and 31 had embryos transferred and or frozen. For PNT treatment, we've, as Bobby mentioned, we have to get case-specific HFEA approvals, and 32 women had obtained approval to undergo PNT. 25 of them have so far had ovarian stimulation, and they've had their eggs retrieved and vitrified. And of these, 22 patients have had eggs removed from storage to perform ICSI, that's the fertilization step. And of the 22 cases who have had an ICSI, 19 have undergone at least one pronuclear transfer procedure, and 18 of these have had embryos transferred and are frozen for future use. So just to talk you through the sort of embryology steps, the most sort of remarkable thing here is that we had reduced fertilization for the PNT patients. So if we compare the fertilization after ICSI of PNT compared with donor eggs inseminated with the same sperm and compared with PGT, we see that there's significantly reduced fertilization for the PNT patients, and to the extent that 3 patients had no normally fertilized eggs available for pronuclear transfer procedure. Some of this reduced fertilization seems to be linked to specific variants. So in summary, 19 patients had at least one normally fertilized egg available for pronuclear transfer. We performed 33 rounds of pronuclear transfer altogether, between 1 and 3 rounds per patient, and PNT was performed on a total of 160 fertilized eggs. And so at the PNT procedure involves some losses, mainly due to lysis of the karyoplast you see here. The karyoplast is containing the pronuclei. And so we got successful nucleation and fusion in 80% of the zygotes in which PNT was attempted. And then on the next day, we had the majority of them survive to the next day. But in all of the 160 procedures that were attempted, 122 survived. And this is showing you the number of intact zygotes per patient. So we had, or per procedure, we had 33 procedures altogether. And you can see that 25% had less than 2 intact eggs. And that's not to do with losses. It's because quite a few of these patients had low numbers of eggs. And for comparison, usually we're aiming for 6 normally fertilized eggs in standard IVF ICSI treatment. So looking at embryo development, its development to the blastocyst stage or after pronuclear transfer and PGT, you see here that just over 40%, almost 50% developed a blastocyst, and there is no difference. This is on day 5 and this is on day 6, and there's no significant difference between the pronuclear transfer and PGT patients. And here, these images are showing you the embryos of different grades. This is a top quality, and this is a good quality. These are both created by pronuclear transfer. And if we compare the proportions of top and good quality, actually the PNT embryos had a higher proportion of top and good quality blastocysts. So 80% of patients had embryos available for transfer. That's the case irrespective of whether we had PNT or PGT. And looking at the outcome of the embryo transfer, we had a positive pregnancy test. So we do a pregnancy test normally 14 days after fertilization. And that was equivalent between the 2 treatments. But by the time it came to clinical pregnancy, which is defined as the presence of a fetal heart at 6 weeks, we found there was a significantly lower clinical pregnancy rate for pronuclear transfer. But if we look at patients coming through the door for treatment, 36% of the pronuclear transfer patients had a clinical pregnancy compared with 41% of PGT patients. So there isn't a significant difference between these. So an equivalent proportion of patients get pregnant, but the PNT patients, we have to work a little bit harder to get them pregnant. And miscarriages occurred in 3 of 20 PGT patients and in none of the pronuclear transfer patients. So we had 8 live births after PNT, 4 female and 4 male, with 1 ongoing pregnancy. And then for PGT, we've had 18 live births, 9 female, 9 male, and no congenital abnormalities reported at birth. And then looking at heteroplasmy, we're at based on measurements in neonatal blood. So this table is showing you the pathogenic variant, the maternal levels. There's 3 homoplasmic women in there, well, 4 in effect, and then 3 that had heteroplasmy. And this is showing you here the level in the egg before we did the enucleation. So this is what the mutation load would have been if we didn't do pronuclear transfer. And then this is showing you the level of heteroplasmy for the maternal pathogenic variant in the neonatal blood. And for this first one, it's 5%. And Bobby, I hope I'm not stealing your thunder here, but I thought it'd be important to say at this point that that was undetectable at 18 months. And then this one we had in 3 cases, in 5 cases, there was heteroplasmy was not detectable at birth. And then this was measured by pyrosequencing because that is the accredited technique in our diagnostic lab. But for the low levels, those that were undetectable, we confirmed by NGS, and you can see here the levels are very low, varying from 0.6 to 0.17. So these are very low levels of pathogenic variants. In 2 cases, we had 12% and 16% in the neonatal blood. So those levels are slightly higher than we would have expected. And we are, but overall, the reduction in mutation load was approximately reduced by 77 to 100%, and 6 newborns was reduced by 95 to 100%, and 2 newborns, the mutation load was 77 to 88, reduced by 77 to 88%. And then just to try and understand better what these 2 higher heteroplasmy levels were about, and it's worth stressing that these are well below the threshold for disease. What we wanted to see could it be explained by difference in the amount of maternal cytoplasm that was transferred during the procedure. So as I mentioned earlier on, when we do this, the pronuclear transfer, you can see the cytoplasm surrounding the pronucleus. And in these images, you can see that amount, here's the pronucleus here, this clearer bit, with a little margin of cytoplasm there, here. And the amount of cytoplasm can vary depending on technical features, technical factors. But and Louis scores for this assigns a score to estimate the amount of cytoplasm that's transferred. And these are showing you the scores here, the PNT scores. And you can see for the higher ones here, 2 and 1, were assigned scores of 2 and 1, which is equivalent to scores in cases where there was no heteroplasmy detected. So we cannot readily explain the higher levels by the amount of cytoplasm that's transferred, but it is possible that we get unequal distribution of this cytoplasm, which could result in levels such as 12 and 16%. So in summary, we had 37 women have undergone at least 1 round of PGT. Heteroplasmy in these women range from undetectable to 98%, and 18 babies have been born. And in the babies, we saw heteroplasmy varying from 0 to 7%, and that's based on 10 infants. And these levels corresponded closely with the levels measured in the embryo. For PNT, we had 22 women have undergone at least, that should be 19 women have undergone at least one PNT procedure. 4 were homoplasmic, 3 were had. Sorry, this is quite confusing. We had a mix of homoplasmic, and we had 12 homoplasmic women and 12 women who had homoplasmic mutations and 12 who had heteroplasmic mutations, and 8 babies have been born. Heteroplasmy for the maternal pathogenic variant varied from undetectable to 16% in neonatal blood. And with that, I thank you for your attention.
Okay, so hopefully we're back on my slide. So from the outset, we've involved patients in the design of the care pathway, and even more so in the design of the study to follow up children. The picture here is a focus group organised by the Lilly Foundation, the leading patient organisation in the UK. And that focus group was specifically to consider the research follow-up of children born. The outcome of that focus group was very clear in that they felt it would really be important for children born following mitochondrial donation to have access to specialist care, that the confidentiality of the babies and their families was maintained throughout the process and during any period of follow-up, and that children were not treated like experiments. So this was the research project we devised with financial support from the Wellcome Trust. And in this research project, the data collection begins as really in pregnancy at 7 weeks when the ultrasound scan confirms a viable pregnancy. It continues throughout the pregnancy into postnatal life. The green triangles are points of contact with our nurse consultant, Catherine Feeney, who remains in contact with these expectant mothers throughout their pregnancy. After birth, the data collection leans into an established routine points of contact that children have with health professionals as part of the UK's standard child health surveillance. And that was a direct attempt to make this feel less like experimentation for the children that were born using this technique. What we have introduced at 18 months is a top-to-toe neurodevelopmental assessment, and that's performed in Newcastle. So the children have to make the journey up here to the northeast of England. This neurodevelopmental assessment includes a multi-domain formal assessment of development, including gross and fine motor, cognitive function, social interaction, and language development. Assessments of hearing and vision, where possible, are undertaken by our colleagues in audiology and ophthalmology, respectively. So, as Mary said, we've had 8 babies born at the time of publication, 4 males and 4 females, coloured pink and blue here for ease of interpretation. We had 1 set of twins. Several different mitochondrial DNA mutations were involved in this process, as Mary's indicated what those were. The disease associations really were isolated cardiomyopathy and myopathy, or blindness. You will see from this chart that most children were born by elective cesarean section. The pregnancies as part of the pathway were considered high risk, and I suspect that's what's led to the elective cesarean section delivery. APGAR scores were all very good. The APGAR is an indication of how babies are functioning at the time of delivery, and a score of 10 is maximal. So 5 children subsequently have had no health problems at all. 1 child has suffered a urinary tract infection, where urinary tract investigations have shown no abnormality. 1 child had some self-resolving muscle jerks known as benign myoclonic epilepsy of infancy. No treatment was required for that child, and these resolved spontaneously within a matter of weeks. 1 child did develop some high blood cholesterol and triglycerides, so blood fat was elevated, They also had a complication. A similar complication occurred in the mother of that child during her pregnancy, and we suspect that the child had some fatty liver as a consequence of that, and that led to this dietary restriction in both the mother and the child. Actually led to resolution of that high blood lipid. The same infant actually also had a rhythm disturbance of her heart, which responded very nicely to medical treatment. At present, all children remain well and are meeting their expected developmental milestones. We've only had one child at the time of reporting through the 18-month developmental assessment, and they did perfectly well on that. We're looking forward to seeing the others come through in terms of the 18-month development. And I was going to finish there. Thank you very much.
Okay, thank you, Bobby and Mary. That was amazing. And I want to just congratulate them for all of this long process over a decade of hard work to overcome these legislative hurdles and all of the thoughtful planning that went into this before you even started, and really how carefully and thoroughly you've investigated these pregnancies and procedures and the follow-up. So it's really amazing. So I want to start with a question that came in from before, and someone who read the paper carefully noted that there were 5 women with a MELAS mutation, 3243, and 4 with a MERF mutation, 8344, who participated in the PNT process, and none had a baby, whereas 11 women with Leber's mutations participated in this process, and 4 given birth to 5 babies. So any hypotheses why the women with Leber's mutations had greater success? I know the N's are small. Any comments about that? Yeah.
Can I just correct those numbers?
Okay.
And I'm sorry, my camera doesn't come on. I think because I use Louise's link to join. So I can just say that the actual number of 3243 patients who've had PNT is just 3, and just 3 and just 4 for the 8344. So these numbers are actually very small, whereas we had 11 Leber's patients altogether.
Yeah.
So I think it's just, we can't make any conclusions on that. But, you know, it is notable that the women who came through for PNT with the heteroplasmic variants had really higher levels of heteroplasmy. So there may be some effect there, but the numbers are too small to
I think also, Mary, it may be that the timing of when they came through, that actually some of the 3243 and certainly the MERF women who came through were a little bit older, and they were people that we identified pretty early in the process, but they'd been waiting for this to be available, and so we're at the top end of their reproductive life, I would say. So we have reduced the age as a consequence of some of our early experiences to 35, 36 next birthday.
Okay. So I do want to add here that there are 2 other people who are going to join us on this panel discussion, and they can chime in at any point as well. One is Louise Hislop, who is the consultant embryologist and lead author of one of the New England Journal papers at Newcastle. And the other is Dieter Egli, who's a tenured associate professor in developmental cell biology here at Columbia, and he's been a pioneer in some of the preclinical work on mitochondrial replacement therapy. Okay, so the next question comes from the chat here, and this question about what are the pros of this method, meaning PNT versus PGT, to manually choose non-diseased embryos. So I think that was already discussed, but maybe you could highlight that and emphasize that point for people.
Sorry,
So,
could you repeat?
So look, what are the advantages of PNT versus PGT, and why would you choose one or the other? I know you've already discussed this, but I just think it's good to highlight this point because I think it's a major important point.
Yes, well, I mean, obviously, PGT is the first port of call for anybody with heteroplasmy. unless there's an expectation that they won't have any low load embryos.
Mhm.
And the advantage is that PGT is an already established procedure in ART to reduce the risk of to reduce transmission of nuclear genetic mutations.
Mhm.
And it doesn't require donors, doesn't require egg donors, therefore it's logistically less complex. So people who are suitable for PGT, and that is a requirement of the UK regulations, is that we offer PGT for anybody who may benefit from that.
And in fact, we can only offer mitochondrial donation for those where PGT is demonstrably not suitable.
Great. Okay, and there was a second follow-up question from that same person asking about options for people in the United States. Before I turn it over to the others, I just want to mention that PGT is available in the United States at a couple of centers at least, at Children's Hospital Philadelphia and here at Columbia. So, and maybe Dieter could speak on that issue as well. But PNT is not. So, is this available for people in the United States to go to England or Australia and participate in your studies?
Speaking for the UK, at present, we're not able to offer that to international patients. It is restricted to patients within the NHS.
Mm-hmm.
However, there have been some developments recently with regards to us actually having a policy development for this to be established as an NHS service. And I think once it is an NHS service directly funded as a service for patients, there may be scope for extending that beyond the borders of the UK.
And Mary, can I ask you if there's any opportunity in Australia?
The opportunity in Australia is maybe some way off because we're just at the early stages of.
Okay.
There's a very complex regulatory procedure here in Australia, and we're working our way through getting the licenses and about to be granted our first one just to start the training and preclinical research. Once we get to the clinical trial, that's just a feasibility study to see if clinical implementation of mitochondrial donation is feasible in Australia. And then depending on the outcome of that trial, the treatment will become available. And at that point, I don't know if it will be just confined to Australian citizens or whether it would be more widely available. But initially for the clinical trial, I believe that that's just available to people to Australians.
Okay, so maybe that's a good segue to ask you then, when is your trial going to begin in Australia, and what are your plans for that?
We're hoping by next year, in the sort of later part of next year. First we have to go. Oh, sorry, my video is now fixed, I think. Yeah, first we have to work our way through these licenses, and then we are going to treat 30 cases once and if we get approval to continue to the clinical trial. 30 cases just to show feasibility. It's across Australia. Australia is a very, very, very big place, and so it's quite a challenge getting clinical networks right across Australia and getting referrals from across the country as well. So it is that's being run by John Christodoulou, who's based at Murdoch Children's Hospital. And then the science will be done at Monash University. We'll be doing continuing preclinical research to address things like carryover and reversion. And then we will the treatment will be offered at Monash IVF, which is a partner clinic.
Okay. So maybe you could ask a similar question to Louise and Bobby about what are your next steps in the UK with this program?
So I think, Monsieur, in terms of, as I was saying, we're developing a policy procedure to convert what is effectively research-supported technique into an NHS service delivery, so our National Health Service delivery would then support this from directly budgeted health funds. And that would be that's our next big step, really, to get that confirmation. The follow-up of children will continue, and we still have funding from Wellcome to do that. And I think that it's important to extend the cohort beyond the 8 children that we have already. That is still a relatively small number, and we're still introducing this with a great degree of caution and safety.
Yeah, and as one of the outside people asked before this session, they will you be able to follow them for 20 or 30 years, because that's the age of onset of labors in many cases.
Yeah, so much of the discussion has been around whether these children will develop mitochondrial disease, and I think we've never set out to detect mitochondrial disease because, as I showed on the slides, the vast array of symptoms that are potentially as a consequence of mitochondrial disease, and the fact that this can come on throughout the age spectrum. So you'd have to design a study that lasted 70 years and involved every organ of the body. And in addition, of course, we are taking away the mutated mitochondrial DNA. So the expectation is not one of disease, but more about whether the technique itself is causing any problems, and that the technique itself is safe in terms of clinical outcome. So that's really how this study was designed. Were there subtle developmental issues that might arise at 18 months of age? We are looking at now introducing a 5-year assessment as well to detect even more subtle abnormalities of neurodevelopment.
Okay, great. And Mary, you raised the issue of reversion, meaning the DNA from the mother may repopulate the embryo at a higher level heteroplasmy that was observed, I believe, in one embryo or a child in Greece who had mitochondrial replacement for other purposes. So this is an issue for Mary, Louise, and Dieter. Are you concerned about this reversion?
Reversion was observed in ES cells, and that was complete reversion to the maternal mitochondrial genome or the nuclear donor. And that was reported by Dieter and by our lab and by Shukrat Metalipov. It occurred in about 20% of ES cell lines. Don't fully understand the remarkable finding, don't fully understand what is going on. We haven't seen complete reversion in vivo, during development in vivo. And I think for the kind of levels we've seen so far, the 12 and 16%, I mean, that could be explained by carryover alone, by just unequal distribution. But there may, for reversion to occur, one would imagine that there's going to be a replicative advantage of one genome over another. I don't know, Dieter, do you have anything to add to that?
Not really. I think you've captured it. I think it is amplified in embryonic stem cells, ES cells, that proliferate dramatically in culture over many passages, and it looks like not as much of an effect in vivo, but of course the data are still relatively limited. I think one needs to keep observing it.
Yes.
I have a general question whether any of the women who you approached expressed any ethical or religious concerns about the procedure.
Bobby would be in a better position to answer that, I think.
Yeah, the short answer is no, Michelle, but that's perhaps because they approached us. They do get a full explanation of what's involved in the process in the MART clinic. They've got an opportunity before that in the MRAC clinic to have a brief overview of the techniques involved. Many of the women who are coming forward, for instance, for PGT and for MD to a degree, have had consultation with their religious leaders if it is something that has troubled them, and we've not had problems in terms of people saying, well, I would like this, but I can't under my religious and ethical framework.
Anyone else want to comment on that, or? Um, if not, then I have another question. So Mary, you and Louise have been using pronuclear transfer, and there's an alternative, chromosomal spindle transfer. That's one that Dieter's worked on. So could you explain why you chose PNT and what are the pros and cons of each?
Well, pronuclear transfer was the one, when we started doing this in 2005, I mean, most of the, any animal evidence there was based on pronuclear transfer at that stage. And spindle transfer came later from Dieter and Chuck Metalopov. And you know, I think there are advantages and disadvantages with each. Pronuclear transfer, for example, has to happen very late at night. Spindle transfer is naturally arrested eggs. So in Australia, we hope to try both and just have a comparison. And for example, for the cases I said where there's some concern about failed fertilization, there's a possibility that could be overcome by performing spindle transfer because you already have them in the wild type cytoplasm. But that's something that we will be investigating. Louise, do you have anything to?
Yes, I'll just add that before we were allowed to start treating patients, our regulators, the HFEA, the Human Fertilization and Embryology Authority, had to approve us and license us. And obviously at that point, we'd published in the Nature paper on pronuclear transfer. So when we were translating the technique into the clinical lab, they were reassured, and that was the technique then that we used, was that we had all that experience. We had experienced embryologists, signed off and competent to do the technique.
Dieter, you want to comment?
Sure. I think, the techniques are very close and practically equivalent in many ways, and clearly both work. So that's the wonderful news. There's an option here. And I think which one's chosen is ultimately secondary. One can mention perhaps there are some indeed advantages of both, technical and perhaps otherwise. When you do spindle transfer, you transfer one nucleus or one genome. That could be an advantage. In pronuclear transfer, you need to transfer both. In spindle transfer, there's the slight challenge is that if you transfer that spindle, you may partially activate the egg and compromise the basic state of the unfertilized egg that needs to be what's called metaphase 2 prior to fertilization. And if that's compromised, fertilization also can fail. You don't have that problem in pronuclear transfer because the egg is already activated. So the other one is, of course, if you manipulate an egg, it's an unfertilized egg, it's not an embryo. While if you do pronuclear transfer, you essentially generate two sets of embryos and then you exchange the nucleus. So there are considerations to be made. I don't have a favorite here. I think they're both very valid and excellent technologies that can be and should be, in my opinion, further pursued.
Great, thanks. I think there are several other questions we can't answer today, but because we have to turn it over now to Phil and his panel discussion.
Thanks, Michelle. We want to make sure we leave time for the patient perspective in this. Maybe one last question that I think perhaps can be answered quickly by either Bobby or Mary. Does the donor oocyte have to be sequenced, right, in advance of the procedure to ensure that no new mitochondrial DNA mutations are being introduced?
Yes, is the very brief answer to that.
Yeah,
It's good in its entirety.
Very good. All right, thank you. Again, thank you all. Fascinating discussion, and I'm quite certain we could stay on for a long time, right, and continue to discuss the clinical aspects of this. But in this first meeting, we wanted to make sure to balance that with the patient perspective. If they haven't already done so, I am going to ask our panelists, Sean and Claire, to turn on their cameras and join us. I will make quick introductions here, and then we can get to a little bit of moderated Q&A. Claire, I'll start with you. Claire is the operations manager at the Lilly Foundation in the UK. The Lilly Foundation has worked very closely with Newcastle in the development of mitochondrial donation. Claire has been working at the Lilly Foundation since 2014, but like so many in the patient advocacy space, she has a very personal connection to it. Her son Jacob, diagnosed young with Lee syndrome and unfortunately passed away at 16 months. Since then, professionally and personally, advancing MRT for the mito community has been very important for her. So Claire, thank you for joining. Very sorry for your loss, and we really look forward to hearing your comments on this. Our other panelist is Sean Murray, the CEO of Mito Foundation in Australia. Sean is a longtime advocate in the mitochondrial disease community, deeply involved in the global mitochondrial disease community on many levels. Much to thank Sean for all his contributions over the years. And Mito Foundation, of course, has played a similar role to Lilly Foundation in Australia in advancing the discourse around mitochondrial donation, mitochondrial replacement therapy. And as we heard in the previous discussion, Australia is now at a point where they're advancing towards the clinic. And I know, Sean, there's a program called Mito Hope that you are a member of the executive committee. I'm sure we'll learn more about that and how the patient advocacy side, right, has played a role in all of this too. So thank you both for joining. Claire, maybe I can start with you. Just to kind of circle back from my introductory comments, as a mother who lost a child to Lee syndrome, can you speak to the unmet medical need and why the desire to have a genetically related child is such an important component, right, of advancing this technology?
I think that's the key, isn't it? I mean, when you're faced with something that you never expected to happen in your life, and for me, it was my first child. You just presume that you're going to be able to have children and they're going to be healthy, and then something happens that just completely puts your whole life in a different direction. And for me, it was, I'll always be a mum to Jacob, but I used to call myself an unfinished mother because I never got to do all the things that everyone else got to do. And you're very aware that there are other ways of doing that in terms of, we went, we looked at adoption and that's a wonderful thing to do. But I think intrinsically humans, that's what we're here for, isn't it? It's to have your own children. It's to see your genetics go on and be proud of what you've created, and speaking to many people in this position, it is so important. And whilst there were things available to certain parents, there was this hole, wasn't there, where there was a set of parents where it was these poor women who felt the weight of the world on their shoulders because it was their fault. And this technique has given them so much hope. And and when this paper was released and it hit the news that these beautiful children had been born to these amazing parents and they were going to live happy and healthy lives, it was, for me personally, it was one of the most emotional things. It was just incredible. It was absolutely incredible. And it made all the work that we put in, both from my professional point of view and as a patient, going to the Houses of Parliament, speaking to to MPs, to Lords, everything we did, to support the amazing people like Bobby and Mary and the brains behind it was just so worthwhile. And it was hope, wasn't it? And there's very little hope sometimes when you're in those dark times, and hope's amazing.
I think that is a word that we can keep coming back to. And Sean, I mentioned the Mito Hope program in Australia. Maybe this is an opportunity for you just to kind of talk about that and what that means in the context of how Australia is advancing mitochondrial donation to the clinic.
Yeah, I think it's a mirror image of what Claire explained there. It really is an opportunity or a signal of hope to the community here in Australia and potentially beyond to to see that for the smaller proportion of the mitochondrial disease community for whom this might be relevant, there's direct hope, but indirectly for the rest of the community, the groundswell of excitement and of hope is palpable, and we hear from those members of our community for in that situation where this might not be something that is relevant for them, but what it's done is it's raised the awareness of mitochondrial disease in regulators and legislators and decision makers in Australia. It's raised the profile of it. Others in the community outside their families have heard about mitochondrial disease now, and just that recognition, the barriers of isolation are being broken down as well. So the ripple effects of this situation are significant as well.
Sure. I'm curious to hear from both of you on sort of framing the technology, right? Like if I'm thinking about in the United States and even how UMDF has talked about mitochondrial replacement therapy, it's more technical, right? An expansion of in vitro fertilization, this technological, innovation. Whereas when I look at the materials, on the Lilly Foundation website, it's more accessible, right? Like a battery pack swap. And I wondered what are your thoughts on the importance of how we frame this technology to the broader, right, communities, not just mitochondrial disease, right, but to everyone, to the public, right, in general. How important is how we're framing it? Because we also know there have been terms like designer baby and three-parent baby, right, that almost feel pejoratives of the judging, and I wonder how your respective organizations have addressed this aspect.
Do you want me to go first?
Sure.
Yeah, because we copied you, so please.
I think you're absolutely right, and I think what it right at the beginning, what it was about was arming our community with the information they needed at the right level to be able to talk to people, and not just family and friends, but to MPs. And it is about pitching at the right level. So you're not glossing over some of it, because you can't gloss over the I don't like the phrase three-parent baby, because it's, to my mind, it never has been a three-parent baby. It's a two-parent baby with a little bit of help from a wonderful woman, and that's the way that I like to look at it. But you need to have those conversations and not shy away from them. But like you say, you need to, if I said to one more person, think about it like a fried egg, and all you do is you take the yellow bit out and you just put that in someone else's, the white bit, and that's what you do. And in the white bit, there's this little bit of DNA that you don't, it doesn't make you who you are. So, it's like getting a new kidney or something, that kind of conversation. And we made sure that we had literature available that our families could read, that was available online. We put together letters that they could send to people, to their MPs, and everything along those lines. And just made it so they felt comfortable in having those conversations, because I think that and then it gave them power to be able to influence the people who they needed to influence.
Yeah, Sean, similar, Australia.
Yeah, very similar to the Friday conversations I had with a number of our MPs in Australia. Exactly. And I didn't even realize you were doing the same thing, Claire. I thought I made that one up, but there you go. But I think that what's important is having the communication material that describes this at different levels for different audiences. So we certainly had the deep science, the longer form, more technical information, validated by experts in the field, trusted experts, both in Australia. We relied heavily in our advocacy campaign on the protracted process that was undertaken in the UK with the number of scientific reviews and the outcomes of those and the outputs of those. So that was the hard data and the hard science that we relied on, and then we brought in and coupled that with local experts, and then added to that the patient voice. And I think we can't underestimate the power of the patient voice in this process. because every parliamentarian, certainly in Australia, is getting hit up with facts and data and budget requests day in and day out. And it's their local representatives who come to their office and sit in there and have a meeting and explain their personal circumstances and the impact of these diseases on them and their families, and what possibility this technique of mitochondrial donation offers.
It's a good point,
that really
John.
cut through.
We often talk about the importance, or the absence of patient engagement, right, really holds back therapeutic development, even more so perhaps in a technology like this, right, where it needs to be explained and conveyed at a very personal level as to, you know, the importance of it. I'm curious, have either of your organizations received much public pushback or blowback, right? Of you know, why are you supporting this? You know, is there a negative side to the story, or how have you prepared for that?
I mean, when we were, you know, talking to people, there were people that were completely against it, and I think you have to accept that, because I think if you're against it, quite often it's for ethical reasons. And I think, especially if I mean, there will be people who have walked in my shoes that ethically don't agree with it. But I would say the majority of people who ethically don't agree with it haven't walked in my shoes. And I think what we were very aware of is, and we said to people, is you can't change some people's minds. You can try, but you know, there has to be, you have to respect other people's opinions. But I mean, we had examples. We've got a very specific example of a very specific MP who was adamantly against it, completely against it. She was going to vote no, without doubt. And we introduced her to a member of her constituency who had one little girl with mito symptoms and was you know, she's beautiful and lovely, but you know, her life is very different to her sisters. And when that MP realized that this wasn't just about Poppy who had mito, it was about her sister and her future and what she was going to be able to achieve in her life. And when she spoke to her mum, and her mum explained that this could give her daughter the chance to have her own family, I think it really struck a bell that she was sat in front of a 14-year-old, and this yes and no vote was going to shape the rest of her life. And she changed her mind. And she voted yes. So, you know, I think, yes, you're going to get pushback. Yes, there are going to be people that don't agree. But it is incredible that the personal perspective can change someone's mind.
Yeah, similar experience in Australia, Sean?
Yeah, very little direct, you know, certainly very little direct, significant pushback to our organization. There was you know, wherever there was social media or, you know, media coverage that allowed the public to comment, of course, there were you know, there was discourse that came through there, but nothing that wasn't a structured and you know, professional approach to disagreement in Parliament, in a parliamentary setting, on a debate on a matter. There was nothing, you know, there was nothing significant. And certainly I wouldn't have, our experience at Mito Foundation was such that it wouldn't be something that I would say an organisation like might shy away from in fear of attracting pushback from community. Certainly our experience in Australia, and I appreciate every jurisdiction is different.
Sure. Actually, I may bounce this going back to Bobby or Mary. You know, I mentioned the National Academies and the report that they put out, you know, in the mid-teens, where they suggested a more cautious approach, right, to advancing clinical research in this space with a focus on male embryos. But it's obvious that has not been the route taken in the UK. Was there consideration as to whether this should be more cautiously implemented? What went into that decision?
But it is cautiously implemented because we offer PGT as a first line of treatment, and PNT only to those who cannot benefit from PGT. However, so in relation to the sex selection, yes, this suggestion was around, but in terms of the regulator, I don't know where it went. I don't think we're allowed to do sex selection in the UK. So it didn't happen. And in principle, I mean, offering sex selection removes reproductive choice from these families as well.
Good point.
Yeah.
Bobby, anything to
Yeah,
add to
no,
that?
I agree with that. I mean, I think we're not allowed to do it, sex selection, other than for a very few specific X-linked disorders.
But
So
as you said, this clinical research is really about testing the technology, right, and the safety of it, the efficacy of it, not so much about the germline editing or long-term effects, right? That's the human condition, is health can go wrong for a lot of different reasons. So I was curious as to your thoughts on that. Just
and we also took the view, Phil, that actually this was about preventing disease in the children, and not in the grandchildren.
right.
So it was one step at a time.
Right. So Claire, Sean, maybe we're starting to run short on time. Again, we can talk a lot of about all of this. UK at a different stage than Australia, already in the clinic and really being used. But maybe what are your thoughts as patient advocates on sort of next steps, right? Claire, is it expansion of awareness, right, in the UK? Sean, how do you build? What role do you play in maybe helping to build the first cohort that will undergo clinical testing in Australia? What comes next on your respective journeys?
I think now that the paper's been published and the results have been so positive from our charity's perspective, we hope that more people will think about this as a potential option and ask the question as to, like Mary said, it's not for everybody and it's not the first line, but it is something that they can consider, whereas perhaps before they knew it was available, but they were a little bit reticent about it because of concerns that, like, the follow-up on these babies has shown today that there are no concerns that we're aware of. So I think that's the next part for us, is it's just something that we hope that more people will ask the question whether this could be something that I'm used for. And like Bobby said, as it's, fingers crossed, available in more places, etc., as we move forward, it will become, PGT is something that people talk about all the time. It's one of the things in our arsenal of possibilities.
Yeah,
And
it's
it will
helping the
just
patients to find the right solution for
yeah,
them, right?
absolutely. So on our website, when we talk about fertility options, you've got genetic testing, you've got PGT, and now you've got mitochondrial donation.
Yeah, super. Sean?
Yeah, and you're spot on there. We've got a ways to go in Australia, Mary. We're very fortunate that Mary's come to our shores now to help the program and to help drive that program here. I think the role for Mito Foundation and the community at the moment is to play the role of, we're engaged in the program itself, and our role is to help represent the patient perspectives in the service as it develops, and to support any supporting regulatory and advocacy work that we can do. We've got a series of regulatory hurdles to get through even after the first in-human treatments are applied. Just based on the way our legislation is constructed, there'll be federal legislation that has been changed. After the pilot progresses, and assuming it follows a similar successful pattern as it does in the UK, in order to then bring it into clinical practice, there are a series of state-based legislative changes that have to be made. And we need to make sure that the government funding persists beyond the existing 5-year window, of which we're already 3 years in. So we've got some work to do around advocating on those fronts, to make sure that things progress.
Well, thank you both for your comments. Again, we could talk about this much more, and we intend to talk about this much more in future meetings. I want to thank all the experts who joined us today, as well as the patient advocates. As I said in my introductory comments, this was not an end station in the process, but rather a very early stop on the process of MRT and what it means in the United States. We recognize we have a very different regulatory landscape, political landscape in the United States, but there's a commitment from not just UMDF, but other patient advocacy groups that represent the mitochondrial disease community to play a role in advancing the discourse. And hopefully we reach a point where we're able to talk about successes the same as you've had in the UK and Australia. We will be convening that meeting in the coming weeks. And for those of you who asked in Q&A and outside what role you can play, we very much appreciate that. I think we want to get ourselves structured first, but please know, right, we will need all stakeholders involved in this process to be effective. I think that's already been demonstrated in both the UK and Australia. So with that, we're going to bring the meeting to a close. Michio, thank you so much for your contributions here, moderating as well. I just want to remind Bench to Bedside webinars are a regular part of our programming. This was a special edition because of the timeliness of the papers, but we will get back to our normal schedule here the second Monday of each month at 12 o'clock Eastern Time in the States. The next seminar will be on Monday, September 8th, the role of rehabilitation medicine in the care of children with mitochondrial disease. We hope you'll consider joining us for that or a future Bench to Bedside webinar. Thanks, everyone. Very much appreciate your involvement. Great discussion, and look forward to seeing you soon.